Okay, time to begin. This is my video on photosynthesis, and let's dive into what photosynthesis is. So photosynthesis, as you already know, is a process by which plants produce energy from sunlight, carbon dioxide, and water. So you've seen this before, 6CO2 plus 6H2O yields C6H12O6, which is glucose, plus 6O2, which is oxygen. And this is a pretty nicely working process, except it's not as simple as this equation shows it to you. These things just don't spontaneously combine into glucose and oxygen, because these are relatively stable or low energy molecules. And in the process, you end up with a super high energy molecule. And well, yeah, oxygen is pretty low energy, except it's pretty easily oxidized. Anyway, so there's no way to get in nature to move from low energy molecules to high energy molecules. Like, nature just doesn't want to do that. Nature rather wants to spend energy and make molecules more stable than making them more high energy. So what actually goes into this process is a whole bunch of energy in the form of sunlight and a bunch of enzymes to help catalyze reactions. And it's really not as simple as you throw these things into a container and out pops up glucose and oxygen. So I just want to put that out there. So a lot of energy and a lot of complicated reactions go into this. And photosynthesis gets broken down into two reactions. Photosynthesis can be broken down into a series of processes called the light-dependent reactions, which, as the name implies, are the set of reactions that photosynthesis requires light in order to perform, and the light-independent reactions. Light-independent reactions, which are also known as the Calvin-Benson cycle. Calvin-Benson cycle. And these processes are cyclic, which means that they repeat over and over again as long as they have the inputs they need in order to keep the process going. And these processes are linked, which means that they feed into one another. So the inputs into the light-dependent reactions are obviously sunlight, as we discussed before, and water. That's really all you need for photosynthesis to perform the light-dependent reactions, which is kind of surprising. The other two main workhorses that the CO2 that you need in order to produce glucose is feeded into the light-independent reactions. And these processes don't directly just exchange stuff. What the light-dependent reactions produce is called ATP, which you're already familiar with. It's adenosine triphosphate. It's a high-energy molecule that just transfers energy. And another thing called NADPH. And NADPH is a molecule you've probably not seen before. NADPH consists of an enzyme complex called NADP+, and when you add an electron and a hydrogen ion to NADP+, you get NADPH, which is an electron carrier, which makes it relatively high energy, which helps the Calvin-Benson cycle get the energy it needs to convert CO2 into a 6-carbon chain. It's not even glucose. So you've been cheated your whole life. You're not really you don't really get glucose from photosynthesis. Photosynthesis just produces a 6-carbon chain, which is then later converted into glucose in other parts of the cell. So really, the process of photosynthesis you're going to learn doesn't even doesn't even make glucose as an end product. So let's go into the process of the light-dependent reactions, which is the more complicated part. So before we do that, we have to go into a bit of plant anatomy. So plant cell anatomy, which is pretty simple. So you have your standard plant cell. Here's a cell wall just for kicks. Let me just make this drawing accurate, because it's fun to make things accurate. So here, let me change the colors around a bit to make this drawing colorful and not just all green. So here's your main vacuole. Here's the nucleus stuck up in the corner. Let me switch back to green. And finally, you have the thing we're interested in. They're the chloroplasts, right? Here's a chloroplast. Here's a chloroplast. Chloroplast. We're not really concerned with much of these. We're only concerned with the chloroplast. And the chloroplast looks something like this. It's just a blob. And inside this gigantic blob, there's this complex system of folds, like a really folded up blanket. And the whole purpose of these folded up blankets is to increase surface area so that this organelle can do as much photosynthesis as possible with the least amount of volume. Like, you've seen this concept a lot in biology. Like, cells are small but have a high surface area so that they can perform a lot of processes. So yeah, this is similar. Like in mitochondria, you have cristae, or the folds within mitochondria, that help increase surface area to increase cellular respiration. So this whole process, so yeah, these folds are to ensure that you have as much surface area as possible within a chloroplast devoted to photosynthesis. So anyway, these stacks are called thylakoid stacks. Thylakoid stacks. Here, let me spell that more neatly out here. Thylakoid stacks. And these thylakoid stacks are all interconnected, so it's really just one giant thing instead of a bunch of stacks. This was a misconception I had before. And now these thylakoid stacks are not floating, but situated in a fluid inside the chloroplast called the stroma. And the stroma has some things that we'll talk about. Anyway, let's get to the nuts and bolts in terms of chemistry. So here is the surface of a thylakoid. We're looking downward onto a thylakoid stack. And let me draw in some phospholipid molecules. So this is a phospholipid membrane that encloses the thylakoid stacks. Okay, so this is a phospholipid membrane. I'm too lazy to draw it all around. So yeah, it's all over here, and that becomes important soon. And here are a few peripheral molecules we have to be aware of. So first of all, there's a protein, a very important protein. There's a very important complex on the phospholipid bilayer, which is called a photosystem. And there are two such photosystems. One is photosystem 1. This is photosystem 1, which we'll talk about later, which is kind of surprising, since this is photosystem 1. And this is photosystem 2. In the process of photosynthesis, photosystem 2 comes before photosystem 1 in terms of the process, which might seem strange to you, but that's only strange because photosystem 1 was discovered first. Just like when we were doing DNA replication, we talked about DNA polymerase. DNA polymerase extending the nucleotide chain and replicating DNA. But the DNA polymerase that actually does that is DNA polymerase 3, not DNA polymerase 1, because DNA polymerase 1 works to correct the errors of DNA polymerase 3. So yeah, it's only named 3 because we discovered it later. So in the same way, photosystem 2 was discovered later, but it actually comes out first. Anyway, so photosystem 2 gets struck by sunlight. And sunlight is made up of light, obviously, and light is made up of particles called photons. And these photons strike chlorophyll molecules. This is chlorophyll, not to be confused with carbon, chlorophyll. And when the photon strikes the chlorophyll, well, the photon has a lot of energy since it's light, and because of this, it transfers the energy into the chlorophyll molecules, and the chlorophyll molecules get all excited. By excited, I just mean that they have a lot of energy. And when atoms and molecules have a lot of energy, what they do is that they either spend it by moving around, or they release an electron by moving it to a higher state. So in this case, the chlorophyll molecules release an electron. Okay? These chlorophyll molecules release an electron because they've gotten excited by a photon. And this electron isn't just going to go into space. It goes down a network called the electron transport chain, which is just a bunch of proteins. This is the electron transport chain. Electron transport chain. And as these electrons move down the electron transport chain, they lose energy. Why do they lose energy? Because they pump a hydrogen ion from this side of the from the inside of the thylakoid membrane, which is called the thylakoid space, or the lumen, out into the stroma. So actively, the cell tries to maintain a high concentration of hydrogen ions out here in the thylakoid space, and a low concentration out here in the stroma. So the electron spends its energy in trying to get a hydrogen ion into the thylakoid space. So as the electron moves down the electron transport chain, the electron's energy is used to move the hydrogen ion into the thylakoid space. Now the electron runs out of energy, which is not too bad, since it hits, because right when it runs out of energy, it's transported to photosystem 1, which, like photosystem 2, uses energy from a photon from sunlight to re-energize this electron. And this electron moves over here, and the photon excites the chlorophyll molecules. Excited. It excites the chlorophyll molecules, which help excite this electron again. And when this electron is excited again, it's moved to another area, and this area combines a enzyme called NADP+. It combines it with the electron and a hydrogen to produce NADPH. And this is one of the end products of the light-dependent reactions, and it goes out to the light-independent reactions. Light-independent reactions. So yeah, let's not worry about that now. So now the electron has produced NADPH, and the hydrogen, there's a high concentration of hydrogen ions out here, and there's a low concentration of hydrogen ions out here in the stroma. So the hydrogen ions naturally want to move towards the stroma because there's a concentration gradient. The problem is, because there's charged molecules, they cannot cross the phospholipid bilayer, which rejects charged molecules. So they need a transport protein to work through. And so plants have devised this really beautiful system. Well, animals did it too in mitochondria, but that's another story. So plants devised this really cool protein called ATP synthase. And you know that ATP is an energy carrier molecule in the cell, and it's always being used, so it's always used up. Specifically, when ATP is used up, it's broken up into ADP and P. And these two float around with because ATP is a relatively unstable molecule, so when it's used up, so it tries to give its energy to other things, and when it does that, it disassociates into ADP, adenosine diphosphate, not triphosphate, and that missing phosphate becomes P. So these two things are now separated, and an enzyme called ATP synthase works to push them together. But in order to push them together, again, since you lost energy in breaking them apart, you need energy to put them back together. So in order to put them back together, you need energy, and you get that energy by having the hydrogen ions move through the ATP synthase. And ATP synthase is a transport protein, but it uses the energy of the moving H+ ion to turn ADP and P together to reform ATP. And this is another product of the light-dependent reactions, then it goes out to the Calvin-Benson cycle to work. So now let's discuss the Calvin-Benson cycle. The Calvin-Benson cycle is a pretty complicated process. Even I don't remember all of it, so let me just look it up just a minute. Because the Calvin-Benson cycle is complicated, I doubt it'll be asked in a lot of detail, but so let me just go over the basics of the Calvin-Benson cycle while I look for the exact diagram, which you probably don't even need to know. But the Calvin-Benson cycle is a process by which CO2 is converted into a 6-carbon molecule. Oh shoot, I forgot to mention something. When electrons are a type of matter, right? So if you transfer an electron from the chlorophyll out into NADPH, you're eventually going to run out of electrons, right? But here's a really cool part about the light-dependent reactions. Along with, I previously said that the light-dependent reactions also use water, but I never explained how they use water. So you probably have no idea where these H+ ions came from, and you have no idea where these E- came from, and you were promised that photosynthesis produces oxygen or you'd be dead. So in order to produce oxygen, photosynthesis does this really, really cool thing. It breaks down a water molecule, which is relatively stable, into hydrogen ions, which are out here, which are supplied here by the water molecule, electrons, and oxygen. So, in this non-cyclic pathway photosynthesis, which just means that electrons are not cycled back and forth between the photosystems, water is broken down to supply the electrons that are lost by the chlorophyll, which is really, really cool. I mean, like, you never see that anywhere else in the naturally occurring, because water is relatively stable, and it uses this really cool process called photolysis. Photolysis literally means light breakdown. Light breakdown. And photolysis is a relatively uncommon type of molecular breakdown, so it's a really cool process. So I don't know if you find it cool, but I find it cool. So the water produces hydrogen ions for this, and it produces electrons to replenish the ones lost by the chlorophyll, and it produces oxygen as a byproduct, since oxygen is not necessary. That's how you get your oxygen. Basically, the water you pour down in your garden becomes oxygen gas. Isn't that cool? You pour in water and you get out oxygen gas. So yeah, I forgot to mention that. So now let's go to the Calvin-Benson cycle. I still haven't found the diagram, but as I mentioned before, Calvin-Benson cycle is how plants use CO2 to produce a 6-carbon chain. So you put in ATP and you put in NADPH, and you get out a 6-carbon molecule. 6 carbon just means there's 6 carbons in a chain. Hold on a moment while I try to find this diagram. Diagram. Ah, here it is. Okay. So, let me look at this diagram. Okay, so fabulous phosphate. Alright, now we're back. And the Calvin-Benson cycle is pretty complicated. Okay, so you start with... I'm going to explain this really simply in terms of carbon, chains of carbon first, and then I'm going to introduce the names. So, this is basically a counting problem, so you're going to have to keep all the numbers in your head. But we start with 6 molecules of 3 carbons. 10 molecules of 3 carbons. We have 10 of these. Okay, so we have 10 molecules that have 3 carbons. So right now we have 30 carbons. Okay? So we start with 10 carbons. And we put in energy to produce... Just a minute. Okay, so 10 carbons, 10 3 carbon molecules are converted into 6 5 carbon molecules. So there's still 30 carbons. So we still have 30 carbons. And these are converted by using energy from ATP. So this is actually not just 3 carbons. There's a few phosphate groups attached, phosphate groups. And these phosphate groups are added on by, this is just a 3 carbon molecule, and then you add a phosphate group from ATP. So ATP provides the energy for these 10 3 carbon molecules to become 6 5 carbon molecules. Now you put in CO2. Now you put in the 1 carbon CO2, and you put in 6 of these CO2s. Right? So now how many carbons do you have? You have 36, because you have 30 here, and you had 6, now you have 6 here, so you have 36 carbons. And now, these 5 carbon chains and this 1 carbon are reconverted into 3 carbons, into 12 3-carbon chains. 12 of these. And you have 12 3-carbon chains, so again, you have 36 carbons. Now, you use more ATP in order to convert these 12 3-carbon chains into modified 12 3-carbon chains, so it's still 36 carbons. And in the process of this, you use ATP and NADPH. Okay, and you use ATP and NADPH. Let me just check how much time I'm running. Whoa, this is a really long video. So you produce ATP and NADPH, and these, you have 36 of these, so you have 12 of these chains. And out of this cycle, only 2 of these chains actually exit the cycle. So only 2 of these chains actually exit the cycle, and these chains bind together to form a 6-carbon chain, which is later converted into glucose in some other part of the cell. The remaining 10 chains are reused in the Calvin-Benson cycle. You see this? You see this? You see this? So these are all reused. Now let's put some names into this. So this original molecule is called PGAL, and this molecule is called RUBP. And this is obviously CO2. And RUBP and CO2 feed into this enzyme called ribulose. Okay, so this RUBP stands for ribulose bisphosphate. And the RUBP and CO2 feed into this enzyme called ribisco, or ribulose bisphosphate carbonate, I believe. Something like that. Ribisco. They feed into ribisco. And ribisco does this conversion into 12 3-carbon chains. This 12 3-carbon chains are called PGA, and this is again PGAL. And 2 of these PGAL molecules exit to form a G3P molecule. G3P. Just a minute, is that right? No, they combine to form this 6-carbon chain. It's called glucose 6,1-phosphate. And that's it, basically. This is all that photosynthesis is. It's a pretty complicated process, though, because you have the light-dependent reaction forming ATP and NADPH for the Calvin-Benson cycle to convert basically what it started with down to what it ended with, are almost exactly the same thing. They're just slightly modified, and the slight modification allows the formation of glucose. So this is photosynthesis. If you have any questions, you can ask me. If you want another video, I can probably make one. So yeah, okay, thanks for listening.